We're starting. A couple things before we get started with our digestive system lecture. Two things I forgot to mention from respiratory. And I don't even know, this was in your anatomy, but it wasn't in your physiology when it really should have been in your physiology. Okay. What is this? What's this? Pardon? Bronchial tree, tracheobronchial tree. All right? As we get smaller, what do we lose? Cartilage. But what are these made out of? Smooth muscle and begins with a C, rhymes with smumective. Connective. Okay, that could be elastic. That could be some collagen as well. Okay. Why do you have smooth muscle there? So we can dilate an airway or constrict an airway. If we have a dilated airway, does more air get in or less air get in? More air. Constricted means less air. Okay. Duh, right? Okay. Your central nervous system controls this. Autonomic specifically. We have two branches, sympathetic and parasympathetic. The sympathetic does what to the smooth muscle in those airways?
Relaxes so it can expand.
Sympathetic, relaxes airways. That means the parasympathetic has to constrict airways. All right? Why do I mention that? Because it's weird. If I were to have blood vessels, we know that they dilate and constrict, but sympathetic does what to blood vessels? Constricts. And parasympathetic dilates. Okay? Important distinction to remember. Think about fight or flight. You want more blood, higher pressure, constricting blood vessels. You want more air, larger airways. Okay? So that's an important thing that I forgot to mention. It was in your, do you guys remember it from the anatomy? But it was never mentioned in physiology. So I want to make sure I mention it in here, just in case it might, appear in a test. You're welcome. Okay. Oh, wait.
I got you erasing it.
All right. Can I erase it now?
Yes.
All right, fine. All right. The other thing I wanted to mention, pull up your respiratory slides. This one right here. Okay, I'm not going to make it bigger because you guys remember me talking about this, and I think I don't remember what I said about it. I could go back and listen, but I'm not going to. I said we don't need to know the volumes. All right.
You said you don't need to know the numbers. Residual volume, that's at the bottom.
Okay. You don't need to know the actual numbers. But I'm going to go through this real quick just to make sure that, and use the numbers to make sure the concepts are gotten, because I don't think I explained this very well either. I think I glazed over it. So we have our total lung capacity over here. What do you think that means?
Total lung capacity, total amount of air.
The total amount of air we can put in our lungs. All right? So what would inspiratory capacity mean? I take in my tidal volume and take in as much as I possibly can. All right? If I take that plus the maximum amount I can exhale, that's my vital capacity. So in other words, my vital capacity is what I am fully capable of moving in and out of my lungs. Okay? That would be a vital capacity, that which is necessary for life. Do you need to move that much all the time? No, there's a lot of reserve here. So that's why we call it vital capacity, because it keeps you alive. All right? So then the amount we can't move, this is a vital capacity minus the amount we can't move. That's our residual volume. Okay, the amount that's always left in the lungs that we can't get rid of, that's our residual volume. Residual volume plus vital capacity is total lung capacity. Does that make sense? So all the air in our lungs. All right? So those are capacities, but then we can express them in terms of volumes too. You know what tidal volume is. You know what residual volume is. We talked about, I did my stupid demonstration where the amount of air I can take in, and then I'm not going to do it again because I don't want to cough. The amount of air I can push out, inspiratory reserve volume, expiratory reserve volume. Okay? So all of those, think about how they can be manipulated to give different lung capacities. That is a big hint for something. I'll let you figure out what that is. Does that make sense? Clear? No? If it's not, say so.
Can you say that one more time, please?
Yeah.
Which part?
The very last portion where you said.
Obviously,
Knowing these volumes, like think about what you can move in and out of your lungs, knowing those volumes, how they relate to your total lung capacity and fit together is important. So if I say your tidal volume is 400 and your inspiratory reserve volume is 2,000 and your expiratory reserve volume is 1,000, what's your vital capacity? Okay, 3,400. So things like that. If you have questions about that, come see me. All right, now let's go into digestive system, or how we make poop. All right, so our digestive system, like I said before with other systems, I like to go the A&P route because you go from the start of the system to the end of the system, and that takes you through how the whole thing works from beginning to end. It's a pathway to figure out. All right, I am not going to hold you responsible for how long this is, so this is just a picture that I threw in there because I think it's interesting how long it is, and we fit all that in here. Seriously? Okay. Some people fit more than others. All right. When we talk about digestion, there are two main processes going on. One is mechanical digestion. So that's where we physically break things down from large pieces to smaller pieces. Okay, from larger pieces to smaller pieces. And where's the best place we do that? In our mouth, chewing things up. So if you want to do this experiment at home, take some food, put it in a bag, and just smash the shit out of it. All right, that's mechanical digestion. Then eat it. All right, notice we don't alter chemical composition. That's saved for chemical digestion. So that's the reason why you break it down into such small pieces, is that you need to be able to have enzymes and other digestive molecules get at this food to break it down from, see if you guys remember, what are these things called? Generally speaking. If I put them in a whole category of things, macromolecules into individual molecules. So like starches into glucose, or proteins into amino acids, and triglycerides into fatty acids and glycerol. So things like that. All right, so general overview. We're going to go from here down and out. Okay? And again, notice that this is coming at the same time that it's coming in anatomy too. We're doing the digestive system this week, or at least the abdomen. All right. So another name for our digestive system, at least the tube that goes through our body, and I'm going to posit this to you. Whatever's inside our digestive system is outside of our body. Does that make sense? Just like what's inside of our lungs is the same as outside of our body because it's in contact. Everything that's in from mouth to anus is still technically outside of our body. All right? We absorb it into the blood to get it inside of our body. All right? So that alimentary canal, 4 layers, and these are really important to know because they all have very specific functions that we're going to go into great detail about. Kidding. All right, the first, the inner lining of the tube is called the mucosa. All right, mucosa, what do you think gets produced there? Mucus is one of the things, but the mucosal layer, that's what everything that you put in your mouth comes in contact with, is the mucosal layer. So one of its primary functions then would be secretion. Okay. The submucosal layer is a very thin layer just on the outside of that. So we're working our way out, which contains, actually we'll go into it. That's just on the outside. Third is muscle layer. Okay, you can guess what that does. And four is the serous layer that surrounds the outside and is continuous with the peritoneum. Okay, so it kind of holds everything together. The reason why I held back on talking about this is because I didn't want to get ahead of myself and talk about it too early. Okay, mucosal layer, mostly epithelial tissue. All right, we know that's lining the surface of everything. Some underlying connective tissue, a little bit of smooth muscle, but the function of that we're not even going to get into. Lots of glands, including mucus. We've mentioned that. And its primary function, secretion. But depending on where it's located, it can also take part in absorption. Some places it secretes, some places it absorbs, some places it does both. Okay? Submucosa. Just on the outside of that mucosal layer. Loose connective tissue, holds things together, has glands in it, has blood vessels in it, lymphatic vessels, and nerves. So the submucosa is very much a control layer within the alimentary canal. Lots of stuff going on, and that's how it gets its nutrients as well. Muscular layer, except for the stomach, there are 2 layers of smooth muscle. All right? Do you guys remember in the anatomage table when one group in particular asked me, there was the esophagus, and it said longitudinal layer of muscle. You guys remember that, whoever that group was? You're like, well, what's this? It's one of the 2 layers of smooth muscle in the alimentary canal. The anatomage table differentiates that in the esophagus, but nowhere else. Weird. I don't know. So 2 layers. One is a circular layer going around. So what's that going to do to your tube? press it. And then a longitudinal layer that's running in the direction of the tube. And what's that going to do? Peristalsis, like move things along. Okay. Can also take part in mixing, which we'll talk about. All right, so now the circular next to the submucosa, then the longitudinal layer on the outside of that. In between those two layers, we'll find out, is another set of nerves. All right, part of our enteric nervous system, which we'll get into in a little bit. All right, on the outside we have our serosa, and that is, like I said, visceral peritoneum. Parietal peritoneum would be lining the abdominal cavity, and that kind of holds things together and lets things move around without friction. Probably nothing of that is new. This is where it gets a little bit newer. All right, the way our gut works in terms of motility, stimulating motility, is by these slow wave contractions. All right, those are rhythmical changes in membrane potential that happen automatically. Where else have we seen this already? The heart, right? And what drove the automatic firing of the SA node in the heart? Leaky channels. You said leaky sodium in particular? Okay, so leaky sodium channels. Here, it's leaky calcium channels. Okay, so we have these slow waves, and these are occurring within the smooth muscle itself. They're occurring within the smooth muscle itself. We have these interstitial cells of K-Hall. If you go on Google and press the pronunciation, they're going to say K-Jol. It's not K-Jol, it's K-Hall. This was the guy that discovered them. And although I've already told you my opinion on guys that discover things and name them after yourselves, K-Hall, he's the guy that first showed us what a neuron looked like and drew them, and there are books like this, so it's getting chills. This guy was actually pretty awesome. Still shouldn't have named it after himself. But then I was thinking about it. Other people sometimes name things after the person who discovered them, and they had nothing to do with it. But name them something else. They're technically pacemaker cells. Okay? So what they do is they go on and they will have a leaky channel that lets positive ions in. Okay, imagine this is resting, and this is your threshold, right? Remember resting threshold? And just before it hits threshold, it'll come back down because potassium channels open and leak out, okay? And this is going on over and over and over all the time in the smooth muscle of your gut, okay? So you have changes in potential, but they never reach threshold. If they don't reach threshold, what aren't your muscles doing? Contracting. All right? So why do we have this slow wave going on of potential in our gut that's not doing anything to contract muscles?
Is it for like summation so that like they can all be summed up?
She said summation. So when you add all these potentials together, right, they produce an extra potential. Good idea, but no.
So it's just like primed and ready.
Primed and ready. Right, primed and ready. Because what stimulates them to go above threshold is nervous system input. Okay? Nervous system input. And hormonal stimuli as well. Nervous and hormonal input cause them to get just above threshold, then you fire an action potential. So what we see right here is the wave-like membrane potential. And then all of a sudden you see here, nervous system input, you have a train of action potentials. That's going to cause it to fire and contract. All right? Here we have another wave of action potentials because the nervous system had input. Okay? The more action potentials you have, this is how the nervous system works, right? The more action potentials you have, the stronger the contraction. Okay? Because remember, action potentials don't change in amplitude. They're all the same height. It's a number of changes, a number of action potentials that will make a muscle contract stronger. And that is where your summation comes in. Okay, does that make sense?
You said the leaky channels were calcium or potassium?
They are calcium. Errors in my way. If you guys actually look, let me see. I make notes to myself to remind myself to say certain things. Leaky calcium channels. Okay, but when that nervous system interacts, sodium goes in. So the waves are produced by leaky calcium, but they also have synaptic inputs releasing the acetylcholine. Acetylcholine opens sodium channels. Okay, so that's what gives you the action potentials. It's a sodium.
And then the potassium is coming out. That's what brings it back down.
Right, the potassium coming out brings it back down. So let's say your between resting and membrane is minus 80, and this is minus 40. Calcium will take it up to maybe minus 41, and then potassium channels will open and bring it back down. Okay, so we talked about this. The action potentials are what actually cause the smooth muscle contraction when these are fired. And again, the more action potentials, the stronger the contraction. I will clarify. Do you guys see the problem? Here, I say sodium. Here, I say calcium. I will figure that out for you. Let's go with calcium. And if that changes, I will let you know. That's a mistake I did not catch, and I am sorry.
Cross out sodium on the previous
Yeah, just
slide.
cross out sodium.
But it's not contracting yet with the calcium entry, right?
Calcium entry leads to contraction.
Where does it say sodium?
Prior slide.
Previous slide right here.
Oh, look at that.
So,
So it's not by the action of sodium potassium pumps, it's by calcium entry?
I never said anything about sodium potassium pumps. Okay. We have calcium channels. We have potassium channels. Those allow for the fast movement of ions in or out. The sodium potassium pump is really slow. That's always going on, and that restores concentrations of sodium on the outside and potassium on the inside. Okay, so this does not take part in all that. It's just going to restore ion concentrations. Okay, really weird, but these smooth muscles, we've already said that they operate in terms of that automatic depolarization, similar to heart. Another thing they have in common with the heart is between these smooth muscle cells, we have gap junctions. All right, what do we know about gap junctions? They're big-ass holes in the cell membrane that connect one to the other and allow ions to freely pass through. So if you have an influx of calcium ions into this cell, then you're going to have an influx of calcium ions into this cell, even though nothing was happened externally to this cell, because they're all connected. Okay, so they're going to contract as a unit. We call that a syncytium. Did you guys get my announcement? I think it was Nick. You asked me a question and I answered it via an announcement in Canvas.
Which one?
That's the problem. There's something about the heart.
I think it was about like why we had the pain in the neck.
Yes, the jaw pain. So I was like, somebody asked a question. I said I'd look it up and answer it. That was you that asked that? I thought it was Nick. Oh, okay. Sorry. Because I forgot to write it down. So I'm going to write down this right now to make sure I know to clarify. Okay, I am human. I make mistakes. I freely admit it. Okay, two types of movements. Okay, so now we know basically how the contraction in smooth muscle that surrounds the gut works. What kind of movement does that initiate? We have two major types. One is segmentation. All right, and the way I want you to think of segmentation, like let's say you have food going down into the small intestine, and it stretches the small intestine. That is a signal. That stretching sends a signal that says, all right, we have stretching in here. There's something in this tube. We need to push it along, or we need to mix it up. Here is the mixing up. So you get stuff going into the small intestine, and then you get a wave of contractions that do this. Back and forth. Doesn't move in one direction. In fact, this actually slows down movement along the alimentary canal. Why do you want to do that? First of all, why do you want to mix? To mix everything together, right? And why do you want to mix everything together in a nice mushy paste? You ever opened up a cat small intestine and it's like this white pasty stuff in there? Why do we want to do that?
Easier to digest.
Easier to digest how?
The enzymes can
Chemically, right. The enzymes can have better access to it. So you're mixing everything together in this nice paste that enzymes can get at it, and they're like happy because they're like, oh, I can do my job finally. Okay, this is moderated by the enteric nervous system. All right, the enteric nervous system, those nerves, set of nerves that are within the submucosal layer and between the muscle layers. That's how this works. Actually, these would be the muscle layer, enteric nervous system.
So only within the muscle layers, not the submucosal.
Not the submucosal. Well, we will talk about what those layers are and how they work in a little bit. Okay, then we have propulsive movements, and we call that peristalsis. And this is what everybody thinks of when we talk about movement in the digestive tract. So, again, what triggers that? Distension. We have food entering our esophagus, and what's going to happen? Whoop, 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 wave of contract. Spitting on myself. Wave of contractions moving that bolus of food down. Okay? But the way this works is we get something in the digestive tract, it stretches ahead of the bolus of food. You're going to have relaxation, and that is active relaxation, meaning that it's on purpose. All right? And then behind that bolus of food, you're going to have the contraction. So if you contract here, just behind it, and you relax here, you're pushing the food forward 1 unit, right? Then it relaxes here, contracts here, and you push it again 1 unit, all right? But if you've ever swallowed something that's really dry or really big, and it got caught in your esophagus, you guys have had this before, right? I know everybody has, don't lie. And you can actually feel it slowly going down. That's the peristaltic waves moving it down. Okay? that is moderated by the parasympathetic nervous system. So the parasympathetic nervous system is going to go here and say, all right, let's fire train of action potentials to contract in this area, and let's not fire action potentials so this area can relax. Okay. Another stimulus for peristaltic movement is irritation. So you get something in the back of your throat, let's say a shot of whiskey, and that's an irritant to your digestive system. And your digestive system is going to say, hey, we want to get that down and out of where it is as soon as possible, so it's going to stimulate peristalsis to move it along. Right? And the parasympathetic nervous system also stimulates without these two in some cases. All right? Again, our myenteric plexus is required, so that nervous system within the gut. Atropine blocks it, which means acetylcholine is needed for this to occur, which suggests that it's part of the parasympathetic nervous system. If we did not have the myenteric plexus, and there are cases where some people are born without it, not people or animals, they don't do peristalsis, which is weird. So you know what you do then? Then they take this big drain snake and put it down your throat, and I'm kidding. You guys are like, really? No. Okay. Here's where we're going to talk about those two different nervous systems in the gut. We have intrinsic control of the gut, and that is from our enteric nervous system. All right, enteric. What does enteric mean? Gut, intestines. Okay. Note that it can function independently of the central nervous system or the peripheral nervous system. So what that means is if you cut off all parasympathetic or sympathetic connections to the enteric nervous system, you're still going to have adequate movement of the tube and secretion. All right, two different places. We have the myenteric plexus. This prefix my refers to muscle. Myenteric, muscle in the intestines. The other one is a submucosal plexus. Where do you think we find that? Submucosa. Right. So two different areas. All right. If the I don't care that you know these. I put them there because they may come up. Meissner. You guys ever heard that guy before? Same guy with touch receptors in your skin. Okay. What was I going to say? Oh, I don't know. Then we have extrinsic control, autonomic nervous system. Parasympathetic, remember, in the gut is just the opposite of everywhere else in the body. When we talk about fight or flight, sympathetic, parasympathetic, rest and digest, right? So parasympathetic stimulates the control of the gut through the actions of acetylcholine, that's the neurotransmitter. And sympathetic inhibits the gut, muscle movement, secretions, due to the actions of what's NE? Norepinephrine. Good. Excellent. You guys are smart. I will tell you guys this. I always tell this at some point to my students. You guys, way, way, way smarter than I am. I just have 20 years of experience. All right. So how do we innervate the tube? How do we innervate the tube? We talked about the different parts. Here's the submucosal plexus. Notice that it's in the submucosa. Here's what I was trying to get at. It controls secretion. So if you're in the submucosa, right next to the mucosa, then it's going to make sense that you're going to control what gets secreted by the mucosa, right? Okay. Also helps aid in absorption. Okay, but we're not going to talk about that function, I don't think at all. Then the myenteric plexus, of course, is going to be between the circular layer and that longitudinal layer, and that's going to be responsible for controlling motility. So when you hear submucosal plexus, think secretion, submucosal secretion. Myenteric, muscle contraction. All right, I like this slide. This is a neat slide. Okay, here is our gut. we would call this the mucosal layer, okay, the epithelium. Here is that layer of neurons, the submucosal plexus within the submucosa, and here's our myenteric plexus right here. All right? Now notice in the submucosal plexus directly stimulates epithelial cells, right? Therefore, they're going to help with secretion. They directly stimulate. So when these fire, stimulate, you have secretion going on. All right? Here's your myenteric. What do you notice about the myenteric compared to the submucosal, besides where it's located? There's more. What else? Pardon? I didn't hear what you said. They're all connected to each other. I mean, they're connected to each other here too, but there's a much greater network of connectivity in here. What does that allow you to do, that network of connectivity? Pardon? Recruit more, greater control, more action potentials, greater regulation of how the gut contracts. Okay? Okay. So, you guys have probably heard that our digestive system kind of has its own nervous system. This is what they're referring to. Okay? The enteric nervous system. All right. So, notice that the sympathetic can influence both, and the parasympathetic can influence both. If we cut those out, these two are still going to be able to operate without the sympathetic and parasympathetic. So when I mentioned that those slow waves and that they need input from the nervous system to fire an action potential, that could come from these guys, or it could come from this guy. Okay? It could come from either one of those.
The reaching threshold can come from sympathetic or parasympathetic, and it can come from just the myenteric.
No.
No?
Action potentials can come from parasympathetic. Remember, sympathetic would decrease the number of action potentials. It would slow things down. So it could come from parasympathetic or the enteric nervous system. Right. All right. One more thing on this that I want to mention. What did I say was the stimulus that the gut knows when to contract? Distension. Okay. So that means you have to have sensory receptors that are going up to the central nervous system or directly to the myenteric and submucosal plexi. Okay? So because you have this internal feedback loop, sensory to motor, the enteric nervous system can operate without the input of the peripheral nervous system or central. All right. So where does parasympathetic come from? Most of it is the vagus nerve, which is cranial nerve number 10. Okay, most of it comes from the vagus nerve. So most of our gut activity occurs because of this little vagus nerve, which you guys found in the anatomist table, right? Sacral division, the sacral plexus, pelvic nerves coming from there. That controls the distal colon to the anus, so that would be related to our defecation reflex. So that would be separate from the cranial division, so this would control the lower end. Remember how parasympathetic nerves are set up. Preganglionic, coming from their origin, those are long fibers. And then postganglionic, those are short fibers. And the postganglionic neurons for the parasympathetic nervous system going to the gut are located entirely within the gut itself. Okay? And its job to excite the ENS, enteric nervous system. All right? Note also, parasympathetic also contains afferent sensory neurons. What does afferent mean? Afferent means going towards. Well, what are we going towards? The brain, right? Going towards the brain. Efferent means going away. All right, so these would be sensory or afferent. All right. As far as the sympathetic goes, remember those are short, preganglionic. Most of them are going to come from those thoracic ganglia, that we found in the anatomage table, those set of neurons that I heard some people say was lymphatic system. I think there was a quiz and somebody called them lymphatic system. They look kind of like that, but they are on either side of the spinal cord. So T5 to L2, that's where they would originate from. And they synapse in prevertebral ganglia. Postganglionic neurons, they originate, come out, and they innervate the entire gut. So they terminate in the enteric nervous system. So parasympathetic and sympathetic both go to your enteric nervous system, not to the actual smooth muscle. Okay? Sympathetic decreases activity of the enteric nervous system, so it's going to slow things down because that's what it does, and has a direct effect to inhibit smooth muscle. Go ahead.
I just want to clarify. You said the parasympathetic and sympathetic go to the enteric nervous system. They don't go to the actual muscle of the gut.
The smooth muscle. Correct. But notice that the sympathetic actually does. I know I said both. I got ahead of myself. Okay, go back. When we talked about the heart and we talked about the SA node, remember it had sympathetic going there to increase heart rate, parasympathetic going there to decrease heart rate. Sympathetic also went to the cardiac muscle to increase contractility. The gut set up the same way. Okay, so parasympathetic will go to the enteric nervous system. Sympathetic will go to the enteric nervous system, but sympathetic will also go directly to the muscle. Okay, so when you think of the gut, think of the heart. That's why the way to a man's heart is through his stomach. Or a woman's. Anyone's. I'm hungry. I'm not hungry, actually.
Just think about it more and you will be. But the
You what?
parasympathetic, just think about it more and you will be.
Yeah, maybe. When we talk about control of gastric secretion, you guys will get a whole lecture about pot roast. Where were we? Okay. All right. What? Stupid thing. Stop it. All right. Neurotransmitters involved. Preganglionic, efferent neurons going away from the central nervous system. Acetylcholine. Postganglionic, acetylcholine for the parasympathetic. Norepinephrine for the sympathetic. So you notice that a lot of what we're saying is repetitive. Okay, parasympathetic, long, short, sympathetic, short, long. The neurotransmitters involved: acetylcholine, norepinephrine. If we're talking about parasympathetic or sympathetic. Okay. Okay. The enteric nervous system, on the other hand, okay, excitatory neurotransmitters, primarily acetylcholine, but there's also substance P, there's also serotonin, and in one particular spot we're going to find there's a different one as well. So there are multiple excitatory neurotransmitters within the enteric nervous system itself that excite. Inhibitory neurons include VIP. Does anybody know what that is? Vasoactive intestinal peptide. Vasoactive intestinal peptides. So what's your guess as to what that does? No?
Constricts the blood vessels.
Blood vessels. Has something to do with blood vessels. And nitric oxide. All right, those are both inhibitory neurotransmitters within the enteric nervous system.
Did you say the VIP again?
Vasoactive, vaso referring to blood vessels, vasoactive intestinal peptide.
Wait, what's that now?
Nitric oxide.
Nitric oxide.
I was like,
No. Duh.
no.
Yeah.
Is ACH for the preganglionic both parasympathetic and sympathetic?
Yes. Okay. Sympathetic, parasympathetic. We'd have ACH here, ACH here as a neurotransmitter. In the sympathetic, here we would have norepinephrine. Here we would have ACH again as our neurotransmitter. All right, some reflexes associated with this. We're talking about these sensory input. We're talking about motor output and secretion. There's something that you should be familiar with because this is going to come up a couple times, the vagovagal reflex. Vagovagal reflex. The pathway to this, all right, you guys remember what reflexes are? They are a pathway. You have a sensory stimulation. You go to the spinal cord or central nervous system. You come back and something happens, right? Like the example that we always use in undergrad. You guys remember this? You touch a hot stove, goes to your spinal cord, and you go, ow. Just like that. Let's say something goes into the stomach or duodenum, stretches it. It's going to send sensory neurons to the brainstem, which is then going to send efferent neurons back to the stomach and duodenum, right? We stretch the stomach. The nervous system finds out about it. It sends a signal back down to do something about that stretch, right? If you have something stretching, think about the Herring-Brauer reflex in the lungs. If you have something stretching, your body doesn't like that. It wants to get rid of whatever's stretching it, right? So it's going to produce a reflex that's going to get rid of it. So that's going to control gastric motor and secretory activity. Let's lube it up and move it along. Defecation reflex. Poop stretches our colon, sends a signal to the spinal cord. Spinal cord sends a motor neuron back to the colon and rectum, and we have a peristaltic push. Okay, and we poop. And then there are also pain reflexes. Pain, in general, inhibits the GI tract. All right, so when you're in pain, you're probably not going to poop because you're in pain. You're stressed out. All right, this slide again. Referred pain. All I want you to get out of this is, of course, you know your stomach is right here, so it makes sense that the pain from a stomach would be in that general location. The weird ones, appendix and small intestine, occur right around the umbilicus. Okay? Kidneys. Let's see, kidneys. Here and here. But where are kidneys located? Here and here. Some areas, like the kidneys, that don't have pain receptors in them, have their pain receptors located elsewhere. Okay? And that was a developmental thing. So that's why you have referred pain for some things where you wouldn't expect them to be. Is it important to know why? Not as much. But when you guys have a patient that comes in, they're like, oh, my belly button really hurts. Like, bing! Let's look at your appendix. Let's do the McBurney's point and push down on it, see if he has, no, it's rebound tenderness. Thank you. I knew somebody had. Thanks for having my back. All right. I was going to print this out for you guys, but I forgot it. Then I thought, you know what? You guys are like my heart and soul because you are totally paperless, and I love it. This is going to be a very important table. This is going to be a very important table. Maybe I should have printed it out because you had it next to when. Okay, fine. Fine. You'll have it for Friday. So each one of these hormones we're going to go over in quite a bit of detail. The first four are the ones that you should definitely know a lot of. I have it in green if it's something that increases I have it in red if it's something that decreases. Okay, so that's what the colors mean on that. So I only put it here because it's something we're going to refer back to. And if you guys like, oh my God, what does gastrin do again? Go to the table. Okay, how to read this? Here's your hormone. Here's what stimulates secretion of that hormone. So if you have protein going into the area where that hormone is secreted, it's going to cause the secretion of that hormone, and so on. Distension, nervous system. Where is that hormone secreted from? Okay, so that tells you that's also where the stimulus is going to occur, right? Okay, and what action it has. What's the action? What does the hormone do? Now, by definition, where do hormones get secreted into? Always, without exception, the bloodstream. So when we talk about all of these hormones, think they're going into the blood. So are they going directly back to the organ, or are they circulating first? They're circulating first, so their actions are going to be a little bit slower. All right, that's just something to consider, and how you read the table. All right, those are endocrine. We also have paracrines. What does paracrine mean? Say it.
Glands.
Oh, so endocrines are glands too, right? What does para mean? What does that term para mean? Round, next to. Local. So these things are secreted locally. All right. One of them is somatostatin. It's a peptide hormone. All right. Like the table, these are the things that somatostatin does. So I'm not going to go over it here because we're going to talk about it later when we talk about what it is actually doing. And histamine is another local paracrine molecule. All right. And what it does. Again, we are going to go over these again, but if you have questions, go back to these slides. All right, some of our neurocrines. What's a neurocrine? I'm going to draw, this is my neuron. This is a synaptic and terminal, right? Normally it secretes neurotransmitters, right? And then you have your synapse, and then you have your next neuron, right? Neurocrines will secrete some kind of hormone that either goes into the blood or goes into a local area. So it doesn't go to another neuron. It gets secreted into another area. Okay? Examples of that, VIP. What does that stand for again? Good. GRP, gastrin-releasing protein. Gastrin-releasing protein. This one's going to come up again, I promise. This one won't. Just saying. I'll be honest with you guys. And enkephalins. All right? That won't come up again either. Oh, shit. It's right here. I give you guys credit and then... Well, at least you're reading. Okay. Let's see. All right. We'll go over these next two slides and we'll take a break. All right. Splanchnic circulation. That is the correct way you pronounce that term, splanchnic. I hear people splanchnic, like they mess it up, splanchnic. It basically just refers to your gut again. Components of this circulation, the GI tract, obviously, the spleen, pancreas, and liver. So the tube and the accessory organs for that tube. The feed arteries, in other words, the main arteries that are going into splanchnic circulation, comprise 25 to 30% of CO. What is CO? Cardiac output. So about 25 to 30% of your cardiac output goes to the gut. All right? That's coming from the celiac trunk, which you guys have already started getting familiar with, right? The superior mesenteric artery. So celiac trunk, upper digestive. superior mesenteric artery, upper intestines, inferior mesenteric artery, lower intestines. And by lower, I mean in terms of travel, not physical space. Okay? In terms of travel, like the path that it goes along. All right? Venous drainage. This is going to be really, 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 really important for physiology and anatomy. Everything that goes into your mouth and gets absorbed goes to the liver before it goes anywhere else. Okay? Everything that goes in your mouth goes to the liver before it goes to anything else. So everything goes through the hepatic portal vein. All right? Then it goes into this network of liver sinusoids where the liver can do what it needs to do with the everything that was absorbed. Okay? Take out toxins, detoxify drugs, break them down into other forms, store fats, release fats, things like that. Has to do that before it gets put back into circulation. What happens if all that stuff gets directly put into circulation? Could be toxic to us, actually. So, hepatic portal system, really important to understand. This is what it looks like. All right? So, from your spleen, what does your spleen do anyway? I didn't hear you.
On my side, it just hangs out. But
Just hangs out? It does. You can live without it.
Immune response?
Somewhat immune response. What else? An even more important function that's related to digestive. Go ahead. Cycles red blood cells, right? So it breaks down red blood cells, splits them into their component parts, sends all of that to the liver to be taken care of. Okay? So that is kind of a digestive function. All right, so it goes in here. Then it comes back to our hepatic veins, into our vena cava, back to the heart, to the lungs, and then out to the rest of the body. Okay? So when they talk about, and you probably talk about this in pharmacology if you haven't already, first pass drugs. No? You take them orally. They get processed in your liver, first pass, then they circulate through your body. So they don't become active until they actually get into your liver. If I'm wrong about that definition, tell Professor Shukair and let her chew me out. Okay? Let's take a break because I need it. And we'll move on.
like 5 minute break?
Yes, 5-minute break. All right, so just to bring everyone else in on the loop, there was a question about the exam. That is on Monday. Right? Content-wise, about a third each topic. So there's 60 questions. About a little bit less than 20 on respiratory, a little bit less than 20 on heart, and a little bit more than 20 on digestive. All right, so we finish digestive on Friday, and then you'll have that exam. You will not have a quiz for physiology next week because you have the exam instead. Okay? Now, don't stress. I would never do anything purposefully to screw you guys over. Everything in my exam is something we have covered related to the IOs and things I have vetted over the previous 2 years. I change everything every year based on feedback I get from students, and by feedback I mean answers. Like all the questions I threw out last year, which weren't that many, I'm revising this year and asking them in a better way. So hopefully the concept gets across to you a little bit better. Okay? If it doesn't work, I throw that question out again, and then I'm like, all right, it didn't work twice. Let's get a new question in there. So I'm very much, I told you guys this, I try to give you a fair shot at things. Okay? That's my thing. It may be, it's not going to be easy. I'm not saying that, but it's going to be fair. I promise. And if it's not, I am very open to feedback. Okay? So, I'm like, all right, just calm down. Calm down. Let's talk about it, okay? After your exam, the next day, it'll be Tuesday, in anatomy, you guys will have the opportunity to do a review on your quiz. So you know how that little review you did with Professor Shukair yesterday, was it? Same thing for this class, right? Last year, physiology was a tough exam for people. It's your first exposure to how I test, your first big assessment in PA school. Yes, all right, I understand why you're nervous. I get it. There will be a few errors. I can guarantee that too. But learn from them and get better, okay? That's why you're here.
Professor, on Canvas it says out of 55. Why does it say that if it's 60 questions?
Those are the questions I threw. So I copy courses over from the previous year. I had 60 questions. I either threw out or gave bonus to 5 of them.
Okay.
So your final score may not be out of 60. It may be out of 58 or 54. If it's out of 40-something, I've got a problem. So I'm not going to hold your hand through things, but I'm not going to be a dick either. Okay? So just please trust me. Trust me. I know it's been a week.
Would you say the quiz that we took on physio recently is a harder example of questions?
Your quizzes are going to be much harder than your exams. Now, that being said, let's do some psychology here. Some students are going to go, bullshit. That was not easier than that. That's natural. That's fine. Okay? I can't please everybody. And I'm not going to try. But you will get through this. You will do fine. You're not going to fail basic science. How do I know that? I have 2 years where people didn't fail. You have a shitload of homework that is buffering your grade. And you have these quizzes that you have 3 attempts to do. And I look at the scores, most people get 10 out of 10. You have a lot of buffers in this class. That's on purpose. Okay? So if you fail, don't be like, oh my God, I'm going to fail this class. Look at your score at the end. You're like, all right, okay, I can come back from this. Okay? Does that help any?
Yes.
Yeah.
Okay. Let's get back to talking about poop. All right, control of gut blood flow. Blood flow is proportional to local activity. What that means is wherever the food is sitting, that's where your blood's going to flow to. Why? Because that's going to be the most metabolically active portion of your digestive tract. All right? So after a meal to your digestive system, you have a 2 to 3-fold increase for about 3 to 6 hours, which is why after about a half hour eating that meal, you sit down and you're like, oh, God. Whew. Yes. Especially if you've had a long day of PA school. You ever done that? You go sit in the couch, you're like, I'm going to study. Like, this is going to be a great night. You eat dinner, and you're like, you wake up 5 hours later, like, where the fuck am I? All right. Causes of activity, increased blood flow. Remember, we have vasodilator hormones that will open up blood, allow more blood in there so that the tissue can be more metabolically active. That's the reason why you have that increased blood flow. Vasodilator kinins. Kinin is just one of these molecules that vasodilates. And low oxygen, all right? That does not have to be related to digestive activity. If your gut is low in oxygen, vasodilators are going to be released in response, so it gets more oxygen. That happens everywhere in your body. Okay? So, this is just saying how we cause increased blood flow to the gut. Nervous system control of blood flow, same thing. Parasympathetic nervous system increases gut activity, therefore you have an increase in blood flow. Makes sense, right? Sympathetic nervous system directly decreases blood flow. Makes sense. All right. When would the sympathetic nervous system be active to increase blood or decrease blood flow? Exercise. Shock. Right? If you're losing blood or you have a drop in blood pressure, you want to basically close off your blood flow to the gut so it can go elsewhere to keep you alive. Your gut's not as important as your heart, right? Or your brain.
So 25 to 30% of blood goes to intestines. So when exercising or in shock, how much does that decrease to?
5 to 10%. There's this other thing called autoregulatory escape. It's kind of a weird concept. So let's say that you have sympathetic nervous system stimulating constriction of arteries going to the gut. There are local factors in your gut that will override that sympathetic nervous system to restore gut blood flow. Okay? It's escaping that regulatory mechanism. That's what that means. I only mention that because it's there. I will not test you over that.
Just auto-regulate escape. You won't test.
I won't test you on that, no. All right, countercurrent oxygen loss. All right, this is a villi or a microvilli within your digestive system. And what I want you to notice about this is you have blood going in. Here's an arteriole. And notice these capillaries coming off the arteriole. But that arteriole is directly connected to a venule without going through capillaries. Anytime you have that, what did we call that in the lungs? When blood flows through but no gases get exchanged? Rhymes or begins with S, rhymes with good. Woo! I almost. Yeah, that would have been a bad word to say. All right, so because you have this shunt, 80. So we can have fun in here, right? 80%, because of this setup, 80% of the oxygen just goes right from artery to vein, never gets exchanged. So what that means is we don't have a very high blood flow to our gut. It's not very high. There's a lot. There's a huge surface area. That is not harmful under normal conditions. That's how it's supposed to be. It operates. We're all fine, right? We're sitting here. We live 70, 80, 90, 114 years. I only say that because I saw somebody on Facebook the other day. She's like her 114th birthday. I was like, good for you. And she looked pretty good, too. If I lived that old, I'm skydiving. Without a parachute. However, if we have circulatory shock, means blood pressure falls for whatever reason, just drops. Decreased cardiac output, let's say heart failure. Hypotension, mechanical obstruction of blood flow. Because we're already only having 20% of our oxygen going to the gut, any major insult is going to just cut off blood flow, oxygen supply entirely, and villi start dying. Okay, notice this is happening in villi primarily. So villi stop dying. If villi die, you can't absorb as much. All right? As you get older, what starts happening to your cardiovascular system? Doesn't work as well, right? You get heart problems. You get hypotension. Villi start dying off. Old people can't absorb as much food, right? Have you heard that before? So this is one of the reasons why. All right, here it goes. We're starting at the mouth. All right? Starting with our mouth, our cheeks has muscles of expression and chewing, buccinator muscles, right? We have our lips. They contain skeletal muscles and sensory receptors. So think of a little baby. You put the little baby right next to a bottle or a breast, rubs. What does the baby do? That's what lips are designed to do, right? Tongue, all right. Tongue has many functions. Mixes food, has taste buds, so we actually enjoy what we're eating. If everything tasted like cardboard, would we want to eat as much? No. Which makes me want to make everything taste like cardboard sometimes, because as you get older, you can't control your weight as easy. And we have tonsils and lymphatics that when we're, because, God, 5-second rule, right? You pick it up, you're still going to eat it. All right, depends on where it actually lands. You put it in your mouth. You want to have protection, right? So the tongue has lymphatic tissues that detect any foreign invaders and mount an immune response before it can get into your digestive system. Oops. All right, saliva. Saliva. Its job to moisten and solubilize food. We can't make anything soluble if it's not wet, so saliva makes it wet. Okay. Saliva contains bicarbonate ions. We talked about that in the respiratory system. What do bicarbonate ions do? They buffer acid. All right. So wherever you see bicarbonate ions, anywhere in anatomy, no, you won't see them in anatomy, physiology, they're a buffer. All right. They buffer against acids, particularly in the mouth here. Your saliva also has oral hygiene effects. We have enzymes in there that break down things. We have thiocyanate, which is, I put that in there just because it's curious. It's a chemical that related to cyanide that actually kills living things, okay, bacteria. And it turns out that the more you smoke, the more thiocyanate you actually have in your saliva, because of the cyanide toxins that's in tobacco. So, yeah, it's kind of gross. And you have antibodies. All right, so, you know, if you're one of these people that's constantly like moving your tongue and moving saliva around your mouth, your teeth are probably going to be in much healthier condition than somebody who doesn't do something like that, right? Because saliva has a very protective effect for oral hygiene. That was really weird. Okay. Now, saliva, it moistens and helps to solubilize things. It does that through two types of secretory cells. And notice that you secrete about 800 to 1,500 milliliters of saliva a day. Where does most of that go? Swallow it, right? 1.5 liters of saliva. Ew. You ever done that experiment where, God, what do you, I don't even remember what you do it for, but you spit into a cup, and I think you're testing amylase activity. Yeah. You guys ever done that in like an undergrad lab? And then you spit in a cup, and like some people are like, I can't spit anymore, my mouth's dry.
Think of a lime.
What'd you say?
You think of a lime or a lemon.
Yeah.
They sell it.
Or pot roast. All right, two types of cells. One is serous cells. Anytime you see the word serous, think watery. All right, so you have a watery fluid. It contains amylase. Amylase breaks down starch, breaks down carbohydrates. So you take big, long, complex carbohydrates, break it down into smaller chunks. That's what amylase does. All right, so much that it converts them into disaccharides, two sugars. All right, so does all of your carbohydrate, like you eat a potato, is every last bit of that potato broken down by amylase? No, because you only have amylase from here down into your stomach, where amylase gets destroyed by stomach acid and pepsin. So it starts the process. So, important concept. Digestion starts where? Mouth. Which macromolecule does it start with? Carbohydrates. Okay? Also secretes mucus cells. Mucus cells, of course, we know is that thick kind of stringy stuff that's in your saliva. Its job is to bind and lubricate. So all of that food that you chewed up, you put it together in a ball that you put in the back of your throat, called a bolus. So that mucus helps bind all that together and makes it kind of slippery so it goes down your throat a lot easier. I like to call it food lube. All right, so where are your salivary glands? You have your parotid glands here, right just anterior to your ear. You have your submandibular glands here, right underneath your mandible. Okay, and your sublingual just under your tongue. 90% of your saliva comes from your parotid and your submandibular glands. Okay, the parotid glands secrete a watery fluid that has amylase, so that's where your breakdown products are going to come from. And the submandibular glands secrete that mucus. All right, so parotid, submandibular, 90% of the secretions. Okay? All right, important but weird. So the sublingual then is the rest of the 10%, and most of that is thick mucousy fluid. Okay? The innervation to our salivary glands comes from sympathetic nervous system. So and this is kind of weird, right? Because what does sympathetic nervous system usually do in a digestive system? Decrease things. Here, because of the way the wiring's set up, sympathetic can stimulate small amounts of mucus. Okay? Mostly it's a viscous secretion, so it's probably mucus, and it's probably coming from sublingual glands. Why does it do this? Why would the sympathetic nervous system do this? When you're...
Get it out of your mouth.
To get what out of your mouth?
Like, because sympathetic is like fight or flight,
Mhm.
so like it wants to. Never mind.
Oh, I see what you're saying. I see what you're saying. No, that's not it. Think about any time you've been stressed out and you're like, I got a really dry mouth, but it's not completely dry. You don't want everything to dry out. So the sympathetic will secrete a small amount to keep your mouth moist enough so that things don't die and functions don't go away. Okay? Most of your secretions come from parasympathetic. All right? Large volumes of watery saliva that have a lot of amylase in it to break down carbohydrates. All right. So, how do we regulate this? And we'll talk about one weird thing here. Okay, so a lot going on in this slide, but what this is saying is in green, things that increase the nucleus in the medulla to increase salivary secretions, and things that decrease salivary secretions. All right, so what's in here? Conditioned reflexes. Somebody mentions lime or pot roast. You start salivating. You learn that, right? Or you learn that you like a certain food, so you start salivating in response to it. You smell it, or you see it, or you start thinking about it. It will increase salivation. Chewing increases salivation. It's a mechanical thing. Taste and smell, of course. Smooth objects. Right, this is where it's weird. So if an object is smooth in your mouth, you're more likely to secrete saliva, as opposed to rough objects in your mouth. Right? What I think of when I think of this, you guys ever seen the original, not this stupid Johnny Depp one, Charlie and the Chocolate Factory?
Yes.
Right? Classic, right? At the end, he gives Charlie and his grandpa this everlasting gobstopper. And what did this thing look like? It looked like it looked like, what do we call those things? The jacks, like the balls in the jacks. It looked like a big jack. And they stuck that in your mouth. Now, if you stuck that in your mouth, would that be very appealing or appetizing? So think about that. Rough objects don't stimulate saliva. But smooth objects do because they're more appealing. They slide around in your mouth a lot easier. Okay? And fear. Like I said, think about fear responses when our mouth gets dry. Okay, so we don't secrete as much saliva. That'll stimulate cranial nerve 7 and 9, which is your facial nerve and your glossopharyngeal nerves, to stimulate salivary glands to secrete mucus. Not mucus, saliva. Okay, that's all of this is saying. Sympathetics can do it directly. Okay, now, if you have sympathetic and parasympathetic going on both at the same time, sympathetic also constricts blood vessels. And if you don't have as much blood going to the salivary glands, you can't produce as much fluid. So they can kind of work against each other too. Right? Just kind of an interesting curiosity. All right. So how does this work? I put arrows in here. We have our tongue. Let's say we have taste stimuli. Notice that the arrows are going this way. Anytime you see something like this, that represents a synapse. So from the tongue, you have sensory input. You go to this salivary nucleus in the medulla. All right? That will send signals out to your salivary glands to start secreting saliva. All right? So a sensory input, bunch of synapses, a motor output. All right. Now, we've talked about the mouth. Right behind the mouth is the oropharynx, right? Potential space that's connected to the mouth. And then we have our laryngeal pharynx down here. All right? We're not going to talk about the nasopharynx because we talked about the respiratory system, right? Oropharynx, passageway for food and air. Laryngeal pharynx, passageway for food only. We don't want to put air down there. We want air to go down the other tube. Okay, what's the other tube? What's this right here? The opening is called the larynx, then into the trachea. Okay, so we want to keep this area and this area separate. So we're going to talk about the swallowing reflex, how that works, and how we keep food going down one tube and not the other. All right, the fancy name for swallowing is deglutition. Right, and there are 3 phases. So this is 3 phases of the swallowing reflex, if you will. We chew food up, mechanical digestion. We mix it with saliva, food lube, and then we roll it into a bolus. Now, I love giving this lecture because I like to make you guys go, next time you eat and you chew up your food, pay attention to what you're actually doing. And you're like, oh my God, I did just roll it up in a ball. Oh my God, I am going and swiping it to the back of my throat. It's kind of interesting. We take these things for granted, but when you pay attention to them, you're like, ew, science. All right. So we roll it into what we call a bolus. So this would be your bolus of food. Now, what do you notice here? Which way is this nerve going? Is it going towards the bolus or away from the bolus? Here's our synapse. So that means it's going away, right? So what's this bolus doing then? Stretching, right? Stretches the back of your throat. So where our uvula would be. All right, so we voluntary phase 1. Chew, roll things up, push it to the back of our throat. After that, it's not so voluntary anymore. We have the swallowing reflex phase. So during the swallowing reflex phase, the soft palate, where the uvula is, goes upward. Why does it go upward? So food doesn't come out our nose. The epiglottis down here, which is normally standing up like this, closes so we don't have food going down our trachea. So that thing in the back of the that bolus in the back of the throat closes off our nasopharynx, closes off our larynx, and then muscles contract to move the food into the esophagus, move that bolus into the esophagus. All right? So, swallowing reflex. Closing off the nasal cavity, closing off the larynx, pushing it down into the esophagus. Now, we have a specific swallowing center in our medulla. Medulla does a lot, right? All right, 3 different areas. I don't care that you know these. In fact, I'll just cross them off. We're going to refer back to them, though. All right, if we look at this, everything over here in dotted lines, those are all sensory going to the swallowing center. Everything coming out from A, B, C, that's all motor. Now, even though it's drawn like this, all of this is on both sides, like all the way around, okay? Does that make sense? So this is just a easy way of getting the concept across. All right, so sensory input from the pharynx and the esophagus, because they're being stretched out, sends signals to the swallowing center. The swallowing center then sends signals back down to the smooth muscle surrounding the esophagus, and that will begin your pharyngeal phase, where the food will start to move down. We'll move past the upper esophageal sphincter. That's what UES means. That will open, and it'll move into the esophagus. That's what this is saying. Okay? Then we go in our peristaltic phase. Based on what we've said in the previous, 50 slides. We haven't gone that far yet. 40 slides. 30 slides? Okay. That was like 37. Can you shut the fuck up already? Is peristalsis something we control? No. All right? It's all smooth muscular activity that's mainly driven by what nervous system? The enteric nervous system. Right? It's mainly driven by the enteric nervous system. So once we get down into the esophagus, we start a wave of muscular contractions with that receptive relaxation that's going to push that bolus of food down. All right? So we divide the esophageal phase into subphases. Like, seriously? Primary peristalsis. So when the pharyngeal muscles contract and push things down, the esophagus then takes over. So that's what primary peristalsis is. All right? A continuation of pharyngeal. Secondary is caused by distension of the actual esophagus. Remember, as food stretches, you relax ahead, you push behind. So the secondary peristalsis would push that bolus of food down until the esophagus is clear. All right. So the upper esophagus, the upper 1/3, is actually striated muscle. What does that mean? No. It's voluntary. So you do have some control over that area. Like, have you guys ever gotten something stuck in your throat and you try your best to work it out? That's contracting those muscles way back there. Like, you can feel them going. Like, alone in your room with nobody watching, do that. Because I don't want you guys to look like an a-hole. I'll do it for you. Lower esophagus, 2/3, is smooth muscle. All right, that means the lower 2/3 is something we have no voluntary control over, and it's just going to go on its own. Okay, this is a video, and I will show this in class because it's interesting. There better be sound, I swear. Really? Nope.
You can do sound effects for us.
No. No, not for this. We'll play it anyway. Fuck it. You guys can watch this with sound. It does have sound, I swear to God. So,
In response to activation of if you create the esophageal loop, you finish the swallow.
Okay, have you guys ever swallowed medication without water and it gets kind of stuck in your esophagus? So you have secondary peristaltic waves going on all the time to try to clear that down there. And then you try to drink, and then some of those just stick and they won't go away. Oh, God. It's terrible. So that would be secondary peristalsis. Going back, that pharyngeal phase, remember I said the upper third of the esophagus is striated muscle. That's because it is controlled by the nervous system, like our peripheral nervous system, whereas the lower esophageal sphincter can be just enteric. All right, so that's why you have different types of muscle there. Okay, so what is the esophagus? Straight collapsible tube, just behind the trachea. Remember those C-shaped rings of the trachea that can accommodate for the bolus of food going down? That goes into the stomach through the diaphragm. All right, so right where the diaphragm is, you would have the lower esophageal sphincter that's normally closed except when a bolus of food is coming down and causes it to relax and open up. All right, that opening in the diaphragm is called the esophageal hiatus. All right, when people have their stomach protruding up through the diaphragm, above the diaphragm in the thoracic cavity, that's called a hiatal hernia. You guys have heard of that before? Hiatal hernia. So because it's going through the esophageal hiatus. Okay, mucus glands line the entire mucosa of the esophagus to help moisten things so food goes down much easier. And then when the food drops in the stomach, this lower esophageal sphincter closes and prevents food from going back up. Now, if you are a person who has acid reflux, usually it's because that lower esophageal sphincter is not working like it should, and that stomach acid and food contents are coming back up, which hurts. All right, stomach. Really briefly, here's your esophagus. J-shaped organ. There's your J. Okay. Let's see. Its job is to receive food and then help with mechanical and chemical digestion. All right. If we look at the muscle layers to it, there's that circular muscle layer that we talked about. There's longitudinal muscle layers. So think about it, squeezing, squeezing, squeezing. The longitudinal would make the J smaller. So you go from a capital J to a lowercase j. And then we have oblique fibers. So what the stomach really does when stuff gets in there is, because it has fibers running in multiple directions, mixes everything up. Okay, that's its job. All right, if we open up the stomach and look inside, there are these folds in there called rugae. All right, rugae are, you guys, have you ever opened up a stomach before, a real stomach, like either in a cat or in a human, real human? Okay. Do what? Most of the cats will have a lot of rugae in their stomach, and usually that stomach is empty, because what rugae do is they decrease the surface area so that it can stretch out, right? So you have these folds that can be stretched out when the stomach is full. So they allow the stomach to expand, and if these rugae are here, it's kind of like a washboard effect. So you're moving food across that washboard and breaking it down mechanically.
Would people who do like competitive eating have more?
Competitive eating what?
Would they have more of that?
More rugae?
Yeah, because they're like constantly like training their stomachs to like eat an absurd amount of food.
I don't know the answer to that question, but if I had to guess, they would probably have less, because they constantly stretching their stomach out so much that they lost their ability to expand, and it's almost permanently expanded. Good question, though. All right, so we have 4 regions of the stomach. We have the cardiac region because it's near the heart. That's actually why it's called that. The fundus, which is this little bump coming up the top. Where else do you find a fundus in the human body? I'll give you a hint. I don't have one. Uterus. Okay? And when you do women's health, this is my most awkward nursing rotation. Woman just gave birth, and she's like, all right, go feel for the fundus. And I'm like, no, dig in there. And I'm like, doesn't that hurt? No, you're fine. Weird. Anyway, it's a bump, all right? When you look at an x-ray, that is where you'll see the gastric air bubble, okay, as a landmark. Why? Physics. Air goes up. Okay? Most of the rest of your stomach is the body, and then we have our pyloric region. Now, our pyloric region is, I'm only mentioning this because this term is going to come up again. We have the pyloric canal right here, and then we have something called the antrum of the stomach. Now, the antrum of the stomach is important because a lot of digestive cells are located in the antrum. So this is where it's going to be located. This is what's called the antrum of the stomach. An antrum just kind of means like an opening.
What was all contained in there? You said
Digestive cells.
Digestive cells that are going to secrete things. All right. Then at the end, we have what's called the pyloric sphincter, which controls food exiting the stomach. All right, and we'll get into how that works as well. All right, couple terms to mention. When you see ORAD stomach versus caudad, ORAD refers to towards the mouth. Caudad refers to towards the tail. So the upper end of the stomach versus the lower end of the stomach. Where the body is versus where the antrum is. All right. What else did I want to say here? Okay, so food obviously is going to get pushed from the ORAD region down into the caudad region. We're standing up most of the time, so food is going to sit here, but then this will contract and push food back up. Remember all those muscle layers will push food back up, it'll mix, come back down. So that's part of the mixing process. And since it's coming from here and pushing up, we call that the antral pump. So it's pumping at this point, it's chyme. It's called chyme. Back up so it can be mixed with digestive juices. Okay? Now, because we've already identified the stomach as something that mixes things and secretes, what are the secretions? We have 2 types of glands that do the secretions. All right? Now, these are glands, but these are exocrine glands. What does exocrine mean? It secretes into a surface or duct. Right, a surface or a duct. So our sweat glands are exocrine glands as well. We have our gastric glands, and this is what our gastric glands secrete. And we have our pyloric glands, which are located in the antrum. And this is what they secrete. All right. HCl, would that be in the stomach or the blood? Stomach. Pepsinogen, stomach or blood? Stomach. Intrinsic factor, stomach or blood? Stomach. Mucus, stomach. Mucus, stomach. Pepsinogen, stomach. Gastrin, blood. All right. Gastrin is a hormone. So by definition, that's going to go into the blood to have its effect. So pyloric region has endocrine and exocrine glands. All right. What are the cell types that secrete these? You guys have probably heard about these gastric glands. If you look at them under a microscope, we call them gastric pits. I don't know if you remember that. It's something we went into a lot of detail in for our undergrads here. 3 cell types. Remember, these gastric glands, these gastric pits, this is where most of them are going to be located, in the body and fundus of the stomach. Okay? So, this is what a gastric pit would look like. So here is that what did you call that, Gabe?
A duct.
A duct. So that would be your duct. And all of these cells that are lining it would be cells that are secreting things. All right? So the 3 cell types that you're going to find in there, mucus neck cells. So that's going to be these. They're going to secrete a mucus that goes directly to the surface. Why they're located close to the surface is because you want to secrete a lot of mucus to protect the mucosal layer of the stomach. Parietal cells, kind of about the middle third of the gastric pit, they're going to secrete hydrochloric acid. That's what gives our stomach its pH, and intrinsic factor. And that's going to be a really important one to remember later on. And then we have peptic cells at the bottom. You might have heard of them previously called chief cells. They will secrete pepsinogen. And pepsinogen is the inactive form of the digestive enzyme pepsin. Okay, pepsinogen is the inactive form of the digestive enzyme pepsin. All right, if we look down here in the pyloric region, 20% of our secretions come from there, from 2 cell types. We have G cells, and those are going to be endocrine. Okay, they're going to release gastrin. Where do they release gastrin? The blood, good. And then mucus neck cells, which are going to secrete mucus and a little bit of pepsinogen. Okay, so this is kind of a decent picture right here that shows you the antral cells versus the gastric cells, and where the cells are and what they kind of look like and where they are in relation to each other. I'm not going to test you on anything like that, just a curiosity. Like when I think about this, I'm like, oh, where are their cells? What do they look like? Where are they coming from? How are they set up? All right. What slide number are we on right now?
47.
47? Damn. I like to do 60 per lecture. Means I'm going to have to hurry my ass up. Gastric acid, 3 major functions. Kills bacteria. Actually, bacteriostatic does not mean kill bacteria. It means slow down their reproduction so that they can be killed by other things. Converts pepsinogen to pepsin, so it converts an inactive form to an active form of the digestive enzyme, and it begins protein digestion. Part 2. Protein digestion starts where? Stomach. With what? And gastric acid. All right. Where does carbohydrate? Mouth. With amylase. Okay. There's a reason why I keep bringing this stuff up.
Why do people get serum tests for amylase? Do you know?
Serum test for amylase?
Yeah. You can test that.
I do. No, I don't remember at the time.
I'm just curious.
But I will look that up. It's a genetic thing. I do know that much.
Okay.
Why specifically? I'm not remembering right now.
I'm just curious. Thanks.
You'll get an announcement. All right, pepsinogen. Pepsinogen. Remember, that's inactive. An inactive form of an enzyme is called a zymogen. Okay. When pepsinogen gets secreted, the way I like to look at this, acid converts it to its active form. So pepsinogen gets converted to pepsin. It cuts off the O-gen. Okay, that's a good way to think about it because really it's a protease. It cuts literally a part of the protein off. So you're cutting part of the protein off. Pepsin is a protein. Once it's made, it can convert pepsinogen into pepsin as well. So that's kind of a positive feedback mechanism. Okay, acid converts pepsinogen to pepsin. Then once pepsins form, pepsin and acid can both convert pepsinogen into pepsin. So it happens a lot quicker once you form the first few molecules. Optimal pH is 1.8 to 3.5. So when you have very little food in your stomach, that's when it's going to be most active. Okay. Once it starts filling up, You got to wait for it to get acidic again before it becomes really active. It gets inactivated pH 5. We'll talk about how pH changes when you eat. And then it can be reversed if it gets to that pH. If it gets up to around a neutral pH, then it becomes inactive and it has no use whatsoever. So that's why antacids can be really bad for you, because you inactivate your digestive enzymes if you take them with food. Like Tums. All right, 2 signals stimulate the secretion of pepsinogen, vagal stimulation by acetylcholine. So your your cranial nerve 10 can directly stimulate pepsinogen secretion. And those cells that secrete pepsinogen can respond to acid. You're like, oh, there's acid. Let's secrete some pepsinogen, because we know what kind of environment we're going into now. All right, anytime I highlight something, what do you think that means? Important, and it's probably going to be on a test. Okay, intrinsic factor. I've already even said that's an important thing to know. The purpose of intrinsic factor, and remember, intrinsic factor is secreted by, go back here, parietal cells, the same ones that secrete hydrochloric acid. Intrinsic factor helps with the absorption of vitamin B12. Vitamin B12 is important for cell division. If we didn't have B12, we wouldn't be able to produce skin cells or red blood cells as fast as we normally do. Which means what for our health? Bad things. All right, so when we take in dietary B12, meat, animal products, it binds to something called R factor. All right, so B12 binds to R factor. Okay, it's in the stomach. Intrinsic factor is also released at the same time. All right, so you have B12 bound with R factor, and you have intrinsic factor, both in the stomach. Okay, they leave the stomach together in the duodenum. Pancreatic proteases digest R factor. If proteases digest R factor, what's happening to B12? It's free. Right? B12 is now free. It's free to bind to intrinsic factor. Okay? So what this says is R factor is important for B12 binding in an acidic environment. Intrinsic factor is important for B12 binding in a non-acidic environment. All right? So then intrinsic factor plus B12, they're in a complex. They go through the small intestine, and they are absorbed in the distal ileum. All right? And the ileum is the last part of the small intestine.
So if you give an IM injection of B12 like into someone's blood, does it still complex with intrinsic factor?
What do you think?
No.
No, because it's going directly into the blood, as opposed to the digestive system.
Can you repeat the acid thing? One's important in an acidic environment, one's like more effective in
Okay, R factor binds to B12 in the stomach. That's a very acidic environment. So R12 is important, or R is important for binding B12 in an acidic environment, whereas intrinsic factor is important for binding B12 in a non-acidic environment. I don't want to say basic because it doesn't quite get basic. It just gets less acidic. Does that help? Okay, cool. All right. And then intrinsic factor, and then B12 is absorbed with intrinsic factor. Intrinsic factor goes on to be recycled. Okay? Intrinsic factor is the only indispensable substance in gastric juice. So you can do without acid, you can do without pepsinogen, you can do without mucus, and you'll live. But if you don't have intrinsic factor, not compatible with life. Okay? So, people who have a disease in their ileum, or have an ileoresection, which means take it out, or have not just atrophic gastritis, you know what that is? Gastric inflammation in the stomach, which causes it to atrophy, okay, which gets rid of the glands that secrete intrinsic factor. What about those people who have stomach surgeries? Like my dad just had a, oh God, what do they call it?